All right. Let's see. So today, Christian Giardina of the Institute of Pacific Islands Forestry will be presenting his research on global warming and carbon cycling in tropical forests, lessons from Hawaii. His research is being done through a permit with the HETF. Please note that permits are required when entering the HETF for research purposes. Christian, you can start whenever you're ready.
All right, can everyone hear me?
Yes.
Great.
Yes.
All right. Well, aloha, everybody. I think I know most people who are in the talk, so that's great. Hoping this can be super informal. If you have any questions, just stop, interrupt me, and take your mute off first, and then we can talk about any of the details. I wanted to highlight the key collaborators on this project: Creighton Litton, Susan Crow with the University of Hawaii at Manoa, Greg Asner at Arizona State University, Paul Selmans with the USGS in California, and Jed Sparks at Cornell University. I also want to acknowledge that the work that is being shared today takes place on the HETF. The HETF sits on DOFA administered lands and represents work between the Forest Service and DOFA to create an environment that's supportive of, conducive of the kind of research that we'll be presenting. Also want to highlight that the lands that both DOFA and the Forest Service collaborate on with the HETF are ceded lands, lands that were taken by force without consent or compensation to the Hawaiian people in 1893, and that these ceded lands continue to represent really important sites and places for the Hawaiian community, and that at the beginning of the talk, I want to acknowledge that we're operating on these lands, and that over the last 10 years, we've increasingly become aware of the importance of acknowledging, but then also working to address ceded land issues in the state, which are largely unresolved. So I'm happy to talk about that topic as well. If anyone has any thoughts or comments or questions, that'd be a great discussion topic at the end. Let me see how to advance here. Yeah, so a big question in climate change is how will carbon sequestration in forests change in the future? We know that the planet is warming. For some areas, it's also getting drier. And the balance of how much CO2 is taken up by forests and then how much CO2 is released by forests gives you a sense for how much carbon will be sequestered by a given forest. These two fluxes are temperature sensitive, and it's this balance that really determines how much a system is going to sequester over time. And I've been doing carbon work for about 20 years, much of that with the colleagues I listed before. And there are surprisingly, despite huge amounts of investment into this simple diagram here, a lot of unanswered questions. So I'm going to go through just a little bit of a primer, so we're all on the same page before getting into the actual research. This gross primary production is basically the total amount of photosynthesis that happens in a forest, and this is obviously not a real tree; it's just a stick figure. And that green object at the top is the forest canopy, and then the brown stick is the trunk of a tree, and this brown part down low is a very simple depiction of below ground, which is the focus of my talk. So photosynthesis, as probably most of you know, is a conversion of light into chemical energy. CO2 and water get together in the leaf and become a carbohydrate and oxygen. And this process, the white arrow, is sensitive to the availability of CO2 and water. As these things go up, photosynthesis can increase. We know that this process is also sensitive to light. And we know that this process is sensitive to chlorophyll, photopigments, and enzyme content of leaves. As those things go up, photosynthesis increases. Gross primary production goes to a number of things, including enzyme activity or supporting enzyme activity. And so enzyme activity is highly sensitive to temperature. And as temperature increases, this photosynthetic pathway also increases. Respiration is the reverse process. It's the conversion of chemical energy into potential energy and heat. And gross primary production, and you'll notice that the green box gets smaller when we talk about net primary production, because about half of what's fixed every year in photosynthesis is released as CO2. And that respiration process has a number of components to it. Some of the drivers of respiration include enzyme content, respiratory substrate supply, how much substrate there is for respiration, and temperature again. And so you can see that there are a similar set of drivers to both photosynthesis and respiration, and that balance represents what we have in ecosystems. That balance can be described as, I see a chat. Oh, okay, got it. Net ecosystem productivity. And so some portion of NPP goes to, can people see my cursor?
Yes.
Or no? Was that a yes, Denali?
I can see your cursor.
Great. So some portion of net primary production goes to building wood, building the canopy, but over the course of a year, and net ecosystem productivity or production is measured on the scale of about a year, you lose some of net primary production to things like senescence of leaves, branches, stems. These are returned to soil as detritus, this yellow arrow here, but also below ground. Most of the detrital carbon is converted back to CO2 during decomposition, but there is some fraction of this detritus flux that sticks around and winds up as soil organic matter. It winds up as forest floor material, and winds up as coarse woody debris. The talk I'm going to give today kind of takes the lid off of the below ground. The above ground piece, we have spent 50, 60 years, in some cases 100 years studying. There's been a shorter window for studying below ground, and studying below ground has a lot of obstacles. One, it's below ground, covered in soil. So all these things that we want to understand are really hard to get to and observe. But there are other complications as well, in that things are connected very tightly below ground through root connections to mycorrhizal connections to soils, this rhizosphere linkage to soil organic matter pool. And then the inputs are complex as well. You have inputs of coarse woody debris and litter from above. You have inputs from roots, from mycorrhizae, and it's a very heterogeneous environment below ground as well, as you go from the surface down to the bottom of the soil profile. But this bottom half of the ecosystem represents some really important action in terms of carbon sequestration. So in this schematic of the global carbon cycle, two numbers that stand out that are worth looking at is the atmospheric pool of carbon at 750 gigatons. That's a lot of carbon. And then the vegetation pool at 610 gigatons of carbon. So there's roughly comparable amounts of carbon in vegetation and in the atmosphere. This is important because small changes in the standing stock of vegetation can influence what happens in the atmosphere. Losses obviously increase atmospheric CO2, and gains through sequestration activities like afforestation increase CO2 uptake, and so carbon storage in vegetation, and decreases the CO2 in the atmosphere. The other numbers that are really important is this on the left side of the graph, this schematic showing that in a given year, there's about 120 gigatons of carbon that are exchanged with the atmosphere between terrestrial vegetation and the atmosphere. About 60 of it comes from the below ground, about 60 of the total flux back to the atmosphere comes from the above ground. And of course, there you have this enormous stock of carbon in soils, which is about twice what atmospheric CO2 carbon storage is. The last number I just want to highlight is this fossil fuel and cement production flux. It's about 5 to 7 gigatons of carbon a year. And the point here is that the flux between terrestrial vegetation and the atmosphere dwarfs by a factor of 20 what is coming out of our fossil fuel industry. And it's not to say that we don't need to think about fossil fuel emissions as being important, just to say that small changes in terrestrial vegetation can have a comparable impact on atmospheric CO2 as the release of carbon from fossil fuel emissions and cement production. So what does Hawaii have to offer in terms of understanding carbon sequestration, climate change, how forests respond to global warming? As some of you may know, Hawaii is become increasingly appreciated for its model study systems in ecology. Peter Vitusik certainly spent a great deal of time understanding and establishing model gradients to look at a range of different questions in ecology. In this study that I'm going to be sharing about Hawaiian tropical montane wet forests, our model system to quantify the impacts of rising mean annual temperature on tropical forest carbon cycling. And today I'm going to be talking about how the input, partitioning, loss, and storage of below ground carbon responds to rising MAT. So big picture, we have a pretty good understanding of climate controls on tree physiology and above ground productivity. Our estimates of above ground forest carbon density are fairly robust and getting more robust. We have really pretty good estimates of soil carbon storage. There's some details that need to be worked out for sure. Things like soil depth need to be better quantified to understand how much is stored in a unit of area. But we've worked hard to develop a pretty good global understanding of carbon storage in soils. We don't know how climate change is going to impact below ground carbon storage, though. There are some enormous unknowns, not even uncertainties, but unknowns. So there are things that you can do to get to below ground work. So this is a project from an earlier life when I was living in Michigan for a few years, between my postdoc at UH Manoa and my current position at the Forest Service. And this project was basically designed to ask the question, how much carbon is stored below ground? And we excavated huge pits and quantified total amount of carbon in roots down to 3 meters. We quantified carbon in soils, and we tried to get a really good picture of how this type of inventory could be used to quantify carbon on a site, and then doing this at multiple sites across gradients, how this method could be used to look at differences below ground. This is not really ideally suited for asking climate change questions, especially if you care about the forest. I have worked on some other approaches to look at kind of more subtle process questions below ground. No need to go into a lot of detail about this figure, but basically using stable isotopes, you can begin to partition what's happening below ground. How much is going below ground from the canopy into roots? How much of that flux going through the rhizosphere ends up as CO2? How much ends up as new soil carbon? How much litter fall ends up as litter layer carbon? And then how much of that carbon is joined by decomposing below ground soil carbon to become the CO2 flux? We can also look at global gradients. This is a paper that Creighton Linton and I published a while back that actually led to this study. And we can kind of assemble an idea of how climate affects things like total below ground carbon flux. So when you assemble data points across the planet, you have a rough appreciation that as things warm up, total below ground carbon flux goes up. You can also plot things like below ground net primary production from across studies on this mean annual temperature axis on the bottom here. And you can see that as temperatures warm up from a mean annual temperature of -5 to a mean annual temperature of about 26, below ground net primary production also goes up. So it sure looks like as things warm up, as mean annual temperature increases, you have an increase in the amount of carbon that is moving below ground and then is being converted from TBCF, total below ground carbon flux, into net primary production. What's also interesting is that when you look at global gradients in soil carbon storage, this is a study by Mac Post et al. It was published in Nature in 1982 and caused a lot of people to scratch their heads. A very large scale study with samples taken from over 1,500 soil pits around the world. What you see is that temperature seems to be less of a driver of soil carbon storage than moisture. So you can have a wet tropical system on the far left, you can have a wet boreal system, and they will store roughly similar amounts of carbon. You can see the very strong effect of moisture on carbon storage going from wet to moist to dry to very dry in the tropics. You can see this pattern very in a simplified way, going from wet to moist in boreal forests. And so what this indicates is that there are things that are happening below the soil surface that really complicate our interpretation of simple things like how much CO2 is coming out of the soil as respired substrate. How do we begin to think about these fluxes as not necessarily being indicative of how much soil carbon a system can store? And that set of slides kind of led us, this group of PIs on the first slide, to start thinking about a system where we could tackle this question in a lot of detail. And really address this simple question: to what extent does temperature regulate below ground carbon storage and below ground carbon processes? The Hawaiian Islands, as a model study system to ask this question, have some really awesome attributes. So Peter Batusik describes a model study system as being an ideal compromise between complexity and tractability, relative simplicity. Our system, all the soils are of basalt origin, and I'll share some more about vegetation. Continuous gradients representing substrate, temperature, elevation, and precipitation. And so we spent a lot of time, and Mike Long is on this talk or in the audience, and he was the person on the ground who actually spent a lot of on the ground time finding these plots. But we worked hard to develop a model ecological study system. We established 9 plots in closed canopy tropical montane wet forests. These plots, this is on the windward side of Hawai'i Island here in this blue box. And it encompasses the wet forest unit of the HETF, as well as Hakalau Forest National Wildlife Refuge that the Fish and Wildlife Service operates. It spans a 5°C mean annual temperature gradient. The gradient is pretty special, globally special, because it has constant vegetation, a constant successional stage, constant soils, constant geology, and constant soil water availability. The vegetation is composed primarily of Metrosideros polymorpha. We all know it as ohia, and also Olapa, Caradendron triginum. I can never remember how to say that. Trignum. Anyone want to help me with that one? Okay. 85% of the basal area is composed of these two species. In working with Greg Asner and his capacity to very accurately measure things like mean canopy height, and so stand biomass from fixed wing aircraft, gave us information to allow us to select the largest biomass plots for each elevation band. And so these are our elevation bands that you're seeing in light blue within this section of forest in this darker blue. And we did this for both maximum height and total biomass. What Greg was also able to tell us is that our plots have stable late successional status. They're mildly aggrading. And so we know that our stands are not in a very early stage of, say, succession after a large scale disturbance. We know that our soils are constant across the gradient. They are made up of this 20,000-year-old tephra-derived acridioxic hydroudand classification. Our radiocarbon work, which I'll show later, also confirms that these are about 20,000-year-old soils across the entire gradient. And looking at soil volumetric water content, we know that the water content of these systems is fairly stable. This is a year-round mean of 12 measurements per plot across our mean annual temperature gradient. You'll see here that there are two dots right next to each other. We have two 16°C plots, one in Waipunalei tract on the far north of the HETF, and then the 16-degree plot in the middle of the HETF. The reason why soil water content is constant across the gradient is while MAT decreases as you go from low elevation to high elevation, MAP also decreases, and the resulting change in mean annual temperature means that actual evapotranspiration also declines as you go from warm to cool. So you have cooling, drying, and a decrease in evapotranspiration that results in this constant water balance across the gradient. This is just a quick schematic of our plot layout where we measure things like litter fall and litter traps. We use these round things, soil collars, to measure CO2. We measure all trees greater than an inch in diameter. And we've been in a pause in terms of measuring tree diameters, but we've been measuring things like litter fall for about 12 years now. We had some core hypotheses driving our work. This is a schematic of how we believe our plot works. And we have gross primary production in this green box at the top. These numbers here are based on literature for this temperature. And so given numbers from previous studies in the literature and also previous studies that we've done, we mocked out this hypothesis with estimates for different fluxes to kind of guide our research. Sorry, there we go. Our hypothesis A is that as temperature increases, as we go from the bottom of the, or the top of the gradient at 13°C to 18.2°C at the bottom of the gradient, gross primary production increases. We see most of that allocation, most of that photosynthate, allocated above ground. There's no change in how much goes below ground in this hypothesis. But we do see an increase in respired CO2, and that's driven by this decrease in forest floor mass and total soil organic carbon. Part of this is also driven by an increase in litter fall that reflects this increase in gross primary production. Our our key numbers are right here. So we have an increase in CO2 efflux as the literature would predict. But in this hypothesis, that increase is driven primarily by this increased decomposition of forest floor and soil organic matter. Our hypothesis B is slightly different. We do see a large increase in TBCF going below ground. Again, these are literature numbers we're using to construct this hypothesis. The key numbers here are in this TBCF, total below ground carbon flux, root and rhizosphere portion of the budget. And this increase that we see in CO2 is derived from an increase in TBCF. And so when we contrast the drivers of increased CO2, the key driver of change on the left is soil organic matter and forest floor, and on the right, it's the increase in total below ground carbon flux. So we used all of our data collecting to test some of these questions. So looking at these two hypotheses and figuring out how to test that, we collected litterfall data, soil CO2 efflux data, and from those data, plus some other measurements I won't get into, we estimated total below ground carbon flux. And what we found is that there's partial support for hypothesis A in that litterfall increases, and that probably did contribute to a forest floor contribution to increased CO2 efflux, which we saw in the middle panel. And we did also find, when we worked out our mass balance, our calculations, that total below ground carbon flux also increased. So partial support for hypothesis A, strong support for hypothesis B. Continuing with our hypothesis test, detrital carbon in the form of coarse woody debris and forest floor declined by about 15 megagrams per hectare per 1 degree C of increased MAT. This decline represents a fairly large release of CO2 from this ecosystem with potential to impact net ecosystem production. Because forest floor carbon declines with MAT, but litterfall increases with MAT, this indicates that forest floor is turning over more rapidly as you get warmer. And that's what this mean residence time of forest floor carbon is indicating, that we go from a mean residence time of about 1.5 years at the cool end of the gradient, and that's cut almost in half when you get to this 18 degree C plot at the bottom of the gradient. We were not necessarily surprised, but we were impressed by how soil carbon in mineral soil really behaved quite differently than this detrital carbon in either coarse woody debris or in litter layer forest floor mass. Increasing MAT had no apparent impact on soil carbon storage. So as you go from 13 degrees C on the left, 18 degrees C on the right, fairly stable. And this stability is reflected in the amount of carbon that we see in 4 layers down to a meter. So this, within the context of our hypothesis test, this data doesn't really support hypothesis A, provides strong support for hypothesis B. Wanted to just touch a little bit on the mean residence time of soil carbon in these different depths. We basically saw no effect of temperature on how quickly these different soil carbon pools at these different depths turned over in response to increasing temperature. So 0 to 10 centimeter soils, basically flat. 10 to 30 centimeter soils, basically flat. 30 to 50, getting more variable, but no clear pattern with temperature. And again, 50 to 90.5 centimeters depth, as you go from cool on the left to warm on the right. No dramatic change, no observable change in mean residence time. You'll notice how this carbon dating method highlights how quickly you go from relatively young carbon in the surface, 0 to 10 centimeters, to very old carbon in the deepest soils, from about 250 on average years for mean residence time in the top 10 centimeters to about 10,000 years in this deepest layer. So thinking about mean residence time in the context of our hypotheses, again, hypothesis A, no support from the data. Hypothesis B, strong support. We wanted to do a little bit more work on this to understand the role of mineralogy. We didn't want to inadvertently exclude the possibility that soil mineralogy had some kind of confounding role on our results. So we looked at short range order aluminum, which is an indicator of mineralogy, especially aliphanes and non-crystalline minerals that have, we know that they have a strong effect on stabilizing organic matter in soil. What we found in the surface is that there was not much of a change, no detectable change in short range order aluminum in the surface 10 centimeters across this 5 degrees C gradient in mean annual temperature. Same for 10 to 30. We did see a significant decline in short range order aluminum as you go from cool to warm for both 30 to 50 and 50 to 90. The reason why this is good to demonstrate, to highlight, is that a higher abundance of short range order aluminum would result in increased capacity of cooler soils to stabilize carbon relative to warmer soils. So if mineralogy was confounding our results, we would need to see an opposite pattern, where the mineralogy of our warmer soils would be higher in allophane content or higher in these short range order aluminum minerals, and these minerals would have to be distributed in the opposite pattern if they were going to have a confounding effect. So actually, the potential is for when you account for the effect of these minerals, that our test is actually quite conservative on this question. And this is simply showing this relationship with depth and short range order aluminum content and mean residence time on the y-axis. And basically it's showing that there's a large scale pattern across all the depths where increasing short range order aluminum does seem to have a strong influence on mean residence time of the carbon in soil. And this is that partitioning by depth as you go from left to right. And so what we can conclude from the study is that temperature-driven increases in total below-ground carbon flux actually drive higher soil respiration, and that it's much bigger than we hypothesized. So when we look at TBCF on the cool end of the gradient, and we look at TBCF on the warm end of the gradient, it's actually a much larger increase than we anticipated. And when we do our below-ground calculations, we find that this very large increase in soil CO2 efflux in an 18 degree C plot relative to a 13 degree C plot is driven almost entirely by TBCF, and then also this increased flux of litter fall through the forest floor. That mineral associated soil carbon below ground, which makes up in this ecosystem a huge fraction of total ecosystem carbon, seems to play a really small role in determining changes that you might see with temperature. A second key finding, oh, I jumped ahead on my finding, sorry about that. yeah, that storage doesn't change with mean annual temperature. And this is also, I think, a really compelling part of the story is that unprotected detrital carbon definitely responds to mean annual temperature. There's faster turnover when carbon is not protected. With that, some take-home messages for the big picture. Storage and turnover of protected mineral soil carbon is relatively constant across our MAT gradient. And that indicates if we are indeed a model system, that as temperatures warm without other major changes, for example, declining soil moisture, temperature by itself may not have the impact on mineral soil carbon that is currently modeled. Storage and turnover of unprotected detrital carbon, however, are very sensitive to warming across our gradient, maybe more so than is currently modeled. And then this last take-home message that the role of soil mineralogy and substrate supply in regulating soil carbon storage and turnover is really significant. We have really just a great team of collaborators who contributed to this, postdocs like Kristen Freeman, visiting scholars like Egbert Schwartz, Kate Heckman, and Chris Swanston, unbelievably stupendous group of technicians, Scott Larson and Mike Long. I think Mike Long might be on this call. Kaimi Moraes, Bernice Huang, who was at our institute and worked at NEON for a while in Colorado and is now doing a PhD on the gradient, the mean annual temperature gradient. Jenny Johansson, Kevin Kenoshiro, Rachel Mosley, Cheyenne Perry. And then students who worked on the project, Jeremy Albano, Darcy Washida, Joey Kitan, Olivia Schubert, Isaac Ito, and Nathan Friday. And with that, mahalo.
Thank you so much, Christian. It looks like, well, we're open to questions now. Looks like Susan has a question.
Yeah, thanks, Christian. That was really comprehensive, and I always learn a lot from your background. So thanks for that. I'm always a student in the carbon world. The question I have, the mean residence time is a really interesting one. Why it doesn't change, but I'm wondering how much species matter for that. So, for example, ohia, as we all know, is a very conservative species that turns things very slowly. If you had a gradient that had invasive species across the gradient rather than native forest, would you expect these results to be any different?
Yeah, that's a really good question. And I know I asked you a hard question when you gave a presentation a couple of weeks ago. And this is, hard questions are appreciated. And so, yeah, I think one of the surprising things for us is that ohia is actually very productive. It has some of the higher litter fall rates and some of the highest TBCF rates that we found in the literature. It's super productive. It just doesn't seem to put a lot of that carbon into diameter increment in the large wood of the forest. And it certainly does, because they get to be very large in size. There's, an analogous system that or an analogous species in temperate forest, sugar maple forests, slow growing, but they have enormous amount of LAI and they produce a lot of litter and they have really high below ground allocation, both standing stocks of carbon in roots and in total below ground carbon flux. So I think changing species could have an effect. I don't know what that might mean if, let's say, litter quality were to be much higher with different species, or if allocation patterns were different, where maybe they allocated a lot more to stem growth and less to, say, foliage and roots. There are other really important questions, I think, with respect to soils that you can also ask, like, would this hold true on sandy soils without any aliphanes? Would this hold true, in other systems? One of the things that we, in a recent paper that we did, is we did a pretty detailed comparison of our HIPNet plot, which is not too far from our mid-elevation MAT plot, with you know, the CTFS Forest Geo Networks data, and we found that, you know, in that comparison that we put in this paper, that amazingly these Laupahoehoe ohia forests are like in the middle of these global gradients in tropical forests in terms of biomass and tree size and maximum tree size and all of these attributes. And you find that these forests are kind of in the middle. They're sort of representative. It's kind of, it was encouraging, and we wanted to highlight that because a reviewer of a 2020 paper that we just published on litterfall, but really a deep dive that I didn't want to go into here. But they said, well, how can you possibly want to publish this in anything except a local journal, because you have a forest that's dominated by a single species, and you're, you know, how representative of it really is it? So that's probably not a very good answer, but the reviewer eventually bought it, and we were allowed to publish it in a pretty good journal. So
yeah, thanks. No, that helps. I think it would be interesting to have, you know, similar information on some of our, you know, fast turnover, you know, species that we get, especially we're seeing that in lowland forest where we're working at KMR. So I was just curious about that. Thanks.
Yeah, and I see Elliot has a really good question. Yeah, that is, I think that's where the state of the art right now is below ground, like really acknowledging that soil CO2 efflux is much more complex than it's really been caricaturized in the literature. And it's been held up as some indicator of ecosystem response to rising temperatures, for example, without really understanding what rising CO2 efflux from soil really means. And as we tried to elaborate in this study and other studies, there are a lot of things that are contributing to soil CO2 efflux. And so your question, I think that's what future studies are going to have to tackle. We don't know, for example, if you warm things up and everything else stays equal, like I was saying, would a sandy soil behave in a similar way to our, you know, very fine textured volcanic soils in Laupahoehoe? But yeah, and that's, I think that's one of the bugaboos too about trying to understand this idea of positive feedbacks to global warming. And for people who don't know about positive feedbacks, the idea is that if temperatures are warming, so as temperatures warm, if ecosystems respond by releasing a lot of carbon, that warming temperatures decompose the carbon in ecosystems more quickly, and that carbon isn't being replaced, that adds more CO2 to the atmosphere. The higher concentration of CO2 in the atmosphere drives more warming, and then that additional warming further accelerates decomposition, and you get into this feedback loop. Conversely, if soil organic matter isn't strongly sensitive to temperature or isn't sensitive to temperature changes, then we can look at other things as potential problem areas for management, or for understanding the implications of warming, say in a feedback framework. So really good question. I think the balance of all the below ground processes and their contribution to the carbon balance of an ecosystem is really important. And we are just, I'd say, at the beginning stages of working that out for a larger area. There's international working group on this stuff, and I'm occasionally a part of conversations, and there's still super contradictory studies that are coming out. One study will show no effect of warming on soil carbon storage. Another will show this enormous impact, but there are all these variables that really need to be, I think, considered more carefully. So one of the things that's amazing about this gradient is that it really is, and the 15 years that we've been thinking about this gradient and working on this gradient, it is the cleanest, most interesting, most highly constrained mean annual temperature gradient on the planet. There hasn't been one like it. So we're super fortunate to have it for asking these climate questions.
All right, does anyone have any other questions? All right, well, thank you so much, Christian. That was an excellent talk, and thank you everyone for coming today.
Denali, I did put something in the chat, and I don't know if we wanted to talk about that a little bit before we sign off.
Oh, sorry about that.
Yeah, it was about the land acknowledgement statement that Christian mentioned at the beginning, and I was just thinking that it might be a good idea for us to open with that at the HETF webinar. I don't know what your thoughts are, Christian, or others.
Yeah, I think that's a great idea.
Yeah, I think it's a good opportunity, and maybe expanding a little bit on the land acknowledgement to talk about what that means and where we are. We can certainly, talk all day about the significance of that acknowledgement statement, but
yeah, great. Thanks for bringing that up. I've been to a number of talks lately at a national level where people are starting to do that, and I think it sets the whole tone for the presentation in a whole different way. So I'm really appreciating that.
For sure. All right. Just like Olivia said, we can definitely do that in our future webinars for sure. Sweet. Yeah.
Thanks everyone for coming. Anyone think of a major hole in our work, something we didn't think about that basically will mean retracting all of our data?
Or gives me another reason to go up to La Boyoe.
Oh, yeah, exactly.
Yeah.
Relocatable, Mike.
Yeah, I just want to say thank you, Christian. It's just a really great presentation. Like working on the project, I kind of knew all these little bits and pieces were also going on, but I haven't seen it quite coalesce or come together in this nice, neat package. So it was really great. Thank you.
Cool.
Yeah.
Wouldn't have happened without you, that's for sure.
That was fun. I miss those days.
All right. Cool. Well, thanks everybody. Really appreciate you joining this afternoon, and eating your lunch with me. And we will see you in the halls or in town. Maybe not you, Jody. When I come over to Honolulu, I'll see you there.
Sounds good, Christian.
All right. Take care, everyone.
Thanks. Bye, everyone.
Thank you. Have a great day, everyone.